Good afternoon, everybody, and welcome to the NC3R's Prize Award event. It's a great pleasure to invite you all. I think we've got over 140 people attending today. My name's Kevin Shakesheff. I'm a professor up at the University of Nottingham, and it's been my great pleasure to start as the board chair of the NC3Rs in the last few months. And today, this prize award is, I'm sure, one of the happiest and nicest occasions that we have as an organisation. Obviously, we're doing this under unusual conditions. I hope everybody is managing well with working at home or wherever you are currently. We're slightly at the mercy of technology, so we're hoping our broadbands all hold up for the next hour or so. So today is a celebration of excellence in science, and we're very grateful for the long-term support of GSK, who have been with us for 15 years now for this prize, which looks to celebrate work from across the world in which excellence in science is contributing towards replacement, refinement, and reduction. We're going to run this as an interactive event, so in a little while we'll be announcing the two winners that we have of the award this year. We'll be hearing from the winners via pre-recorded presentations, and then asking you if you'd like to ask a question to use the Q&A function in Zoom. And at the end of the session, we'll then read out the questions, and we have the prize winners here who will be able to answer those questions. So a little bit about the prize itself. It's very much within the mission of the NC3Rs to promote across the world excellent science. Today, this year has been absolutely a fantastic example, with many superb pieces of work submitted to the International Review Panel, who had an extremely difficult choice to make, looking at outputs that covered the whole spectrum that you might expect of refinement and replacement and reduction opportunities. As I said earlier, it's 15 years since this award started, and we'll hear in a little while from colleagues from GSK who have helped make this prize possible and grow in status. I should also thank the many people at NC3Rs that make this award possible. So a lot of hard work goes into publicising the awards and then processing all of the excellent applications, and then the international panel will come together. And we really looked at the scientific quality of the papers that have been submitted, their potential to impact on the 3Rs, the commitment that the authors were already showing to disseminate that impact to a wide community. And also there's a prize grant associated with the award, and so we looked at how that was going to be used and the benefit that would bring to the future use of the work described in the paper. So we deliberated, and in a few minutes we'll give the results, and we'll ask the prize winners to give their presentations. But I'd now like to hand over to a pre-recorded statement by Dr. Rab Prinjha, who is the head of Adapted Immunity Research Unit at GSK, and is going to say something on behalf of our sponsors.
Well, thank you, Kevin, for the introduction. It's a real pleasure to be here and to represent GlaxoSmithKline in the 15th year that we've been supporting the 3Rs awards. It's a real privilege to represent Margaret Landy and the Office of Animal Welfare within GlaxoSmithKline. To GSK, 3Rs has been a central part of our philosophy, and the reduction, refinement and replacement of animals has been a core part of our R&D strategy, and it's now more than ever incredibly important that we design studies where every intervention, every data point, every animal that's used helps us to advance the science, helps us to help move medicines forward that can help patients in the long run, and 3Rs is a crucial part of that. It's been a real pleasure to look at the applications, and I know that the panel had a really hard time choosing between the winners, and the quality and the breadth actually of the species involved and the types of studies that were done really illustrates how the UK research community is really embracing 3Rs, and really pleased and excited to see the two winners that we're going to announce today. So thank you.
Great. Well, once again, we're really grateful to have GSK work with us so closely on this and on other activities that the NC3Rs undertake. So what I'd like to do now is tell you a little bit about the prize winners before we hear from them. We have two winners this year, and I think that reflects the very high quality of the work that was presented. So the two winners are Dr. Francesca Nunn and Dr. Marta Shahbazi. And I'll say a little bit about both of them in sequence. And they both receive a prize grant, which is for £15,000, and a personal award of £2,000 as well. So Dr. Francesca Nunn is a postdoctoral researcher working in Alasdair's Nisbet's lab at the Moredun Research Institute in Edinburgh. Her paper was entitled "A Novel High Welfare Methodology for Evaluating Poultry Red Mite Interventions in Vivo," and it was published in February 2019 in the journal Veterinary Parasitology. The paper describes the development of an on-hen mite feeding device, and this is used to improve the screening of vaccine candidates and avoids unnecessary field trials. Using this device, they just required 56 hens to test 7 vaccines, whereas the traditional field studies would have used almost 5,500 birds. The new method also allows for much shorter exposure to a significantly reduced number of mites due to the innovative mesh design. And the panel was impressed by how the approach was able to effectively test potential vaccines, obviously a significant reduction in the number of hens, and also a refined protocol that simultaneously improves the animal welfare. So many congratulations to Francesca, and we'll hear from her later, and we can also ask questions through the Q&A function in Zoom. And then the second winner, and just to emphasize this, this is a joint first prize, has gone to Dr. Marta Shahbazi. Marta is from the MRC Laboratory of Molecular Biology in Cambridge, but she's recently set up her own group to investigate the mechanisms of human embryogenesis. Her paper was entitled "Pluripotent State Transitions Coordinate Morphogenesis in Mouse and Human Embryos," and it was published in Nature in November of 2017. The paper demonstrates the ability to develop advanced three-dimensional cultures of both human and mouse embryonic stem cells to mimic the development of the embryo at the point of implantation, and the subsequent morphogenesis and formation of the amniotic cavity can be studied in this in vitro model. This provides a great opportunity to replace and reduce the use of live mice for some of these early developmental biology studies. So the panel thought that this research exemplified very high quality, excellent science, and that this approach could have a very significant impact on animal use and on the insight that you could get into these processes in early embryogenesis. So again, huge congratulations to our two winners. I think they really show the diversity of the type of science that goes into the work that the NC3Rs funds and hopefully the community across the world, and obviously a lot of work in the UK to make this have real impact across industry and academia. So we're going to hear now, back to back, two short presentations from Dr. Nunn and Dr. Shahbazi. And if you're thinking of questions as you're going along, pop them into the Q&A chat, and then Katie's going to come on towards the end and we'll pose the questions to the prize winners.
Okay, good afternoon. I'm Francesca Nunn. I'm really delighted to have been awarded joint winner of the NC3R's International 3Rs Prize this year. And I'm really grateful for the opportunity to share the paper and some of the work that we do at Moredun this afternoon. So the paper is called A Novel High Welfare Methodology for Evaluating Poultry Red Mite Interventions in Vivo. And just a bit of background to give you some context. The poultry red mite is a blood-feeding ectoparasite of hens, or of all birds, but it's a big problem in the egg-laying industry. Now, it has quite a simple life cycle. Eggs hatch into larvae, larvae then transform into protonymphs, but then protonymphs must feed in order to turn into deuterymphs, and deuterymphs feed in order to turn into adults. And in optimal conditions, this life cycle just takes 7 days from egg to egg. Now, the mite is quite tick-like in morphology and in behaviour. It doesn't reside on the host, it just visits the host at night in order to feed. The rest of the time it resides in the sort of machinery and nooks and crannies of the hen house. And you can see in the photos that sort of rust red accumulation. That's all red mite. It's a big welfare issue for laying hens. It can increase stress behaviours such as pecking, cannibalism. It can cause anemia. And in high infestations, it can even increase mortality. Because of that, obviously, it's also an economic issue for the industry, and it also can lead to decreased egg quality and decreased egg production. Control of red mite, because it lives off host, is problematic. So traditionally, hen houses would be thoroughly cleaned out between production cycles and used of chemicals to control the population. Unfortunately, this chemical arsenal is now diminishing, mainly due to food safety issues and due to mite resistance. There are other products that you can put in the drinking water which have an effect on mites, such as Exalt, and this is a synthetic fluoroliner, and it's very effective in the short term. But again, there are worries that eventually you would get some resistance. So the holy grail really in red mite research would be a vaccine. And there are several international research groups working towards vaccines in poultry red mite control. We used to test these sort of novel vaccine compounds using an in vitro feeding device. So you would inject your hens and then use blood withdrawal from them in your feeding device to look at effects on mortality and fecundity. They're quite simple. They usually have a chamber that contains the mites. You must have good mite containment, which is a lot more tricky than it sounds. And then the other compartment, you would have your heparinized blood meal. Usually they're separated by a feeding interface, which will be a membrane, quite often paraffin. The trouble is with the in vitro assays, they're quite difficult to perform, and you get a lot of device failure, especially when you're using a synthetic membrane such as parafilm, because they tend to split when the mites feed through them. You get inconsistent and poor feeding rates and a high background mortality. And all these failures mean that you need a high replicate count, which means more invasive sampling of hens. Often results from your in vitro feeding trials didn't carry through to a field trial situation, which is where you would test your vaccine candidates next. And these usually contain high numbers of birds per experimental group who undergo their long exposure to mites for extended periods of time. So the group were really driven to find a better way, both in terms of hen welfare and to get better data. And they invented this prototype feeding device, which is a soft pouch, and it's made of a nylon phytoplankton mesh. And this attaches to the hen's thigh for 3 hours. And using this, they were able to get consistent feeding rates of about 50%. Now, it gave good containment and recovery of mites, and we think that the good feeding rates are due to the natural conditions and feeding cues. And it also gave a low background mortality. And it's high welfare for the hen. So there's no blood withdrawal involved. And it's really practical to perform. You can have several multiple test groups and replicates if you need them. This was an important step forward in both terms of reduction and refinement. It greatly reduces the numbers of hens required per study. In a field trial situation, you might need as many as 384 birds per test group. In this study, we only require 4. It also will reduce the testing of suboptimal products in field trials. It's also a big step in refinement and improved welfare. It decreases the need for prolonged exposure to high numbers of mites in a field trial situation, and greatly reduces the need for invasive blood sampling of hens. So with the pilot study, the group went to the NC3Rs and were awarded a 3-year grant to further develop the device for the other blood feeding life stages and to see how it acted longitudinally in longer field studies. So when I began, I looked at the same kind of mesh that we used for the adult females in the pilot study. This was phytoplankton mesh. But the aperture size was too big and the proteins could just run through. So we looked at 75 micron aperture mesh and 125. But as you can see from here that we didn't really get much in the way of nymph feeding at all, and adult feeding was quite variable. I then went on to test several polyester meshes. You can see at the top the 68 micron, the 105 and the 120 all denotes the aperture size. But from left to right, the meshes also vary in thickness. So the 68 micron mesh was very thin at 43 microns, and the 105 slightly thicker at 65 microns. You can see the 68 mesh, we didn't really get any protein feeding worth talking about. The 105 worked slightly better. In fact, there was no statistical difference in adult and deuterant feeding between the 105 micron mesh and the 120. However, the 105 was much more successful when it came to feeding proteins, so we chose that one to move forward. We then wanted to optimize feeding rates and also minimize mortality rates, and we did this by means of a conditioning study. When we collect mites from a farm, we then keep them at room temperature for a week. This lets them digest the blood meal they've just had. It allows females to lay eggs and gives time for those eggs to eventually turn into proteins. And it allows the deuterants to molt into adults. We then store them at 4 degrees until such time as we want to use them. So for this study, we used batches of mites that have been stored for various periods of time at 4 degrees and used them in feeding assays. And what we found was that mean adult feeding rates significantly dropped at 2 weeks at 4 degrees, but mortality was lower at 2 or 3 weeks. Mortality in nymphs didn't vary across the time points for either of them, but mean deuterant feeding increased at 1 week at 4 degrees, whereas mean protein feeding increased at 3 weeks at 4 degrees. So we could use this data going forwards into different studies and to test different vaccine candidates. Since then, the on-hen feeding device has been used successfully to assess the efficacy of 7 novel vaccines. 3 of those in our laboratory were in conjunction with 2 other research groups, one at the Royal Veterinary College and one at the Norwegian Veterinary Institute, and 4 with a group with Dr. Jose Lima Barbera in Spain. The device, we were able to determine that none of these vaccine preparations on their own warranted their own field trial. And each of these field trials would have used 784 hens, including controls, so just under 5,500 hens in total. Also, testing the vaccines in this way meant exposing just 56 hens to 100 mites for 3 periods of 3 hours, instead of the nearly 5,500 birds in constant exposure to more than 10,000 mites each for about 100 days. The results of these studies can be found in peer-reviewed journals, 3 of them in vaccine, and there are more to follow. We're also delighted to be awarded an NC3R Skills and Knowledge Transfer grant to hold a 3-day workshop at the Moredun Research Institute. And this will involve researchers from all over the world and have practical hands-on sessions and also discussions on how else the device can be integrated with new technologies emerging in the field of PRM research. Of course, at the moment, this will have to wait until such time it's safe for everybody to travel. I would like to use the grant that we've been awarded in the prize to build upon improvements of reduction and refinement using an in vitro testing device. So despite my criticisms of the in vitro device earlier, there's still a place for one that works well. And last year I had an amount of time in which I tested different feeding membranes and different device prototypes. And we came up with one that has given us really, really good feeding rates, and it's really, really reproducible. The membrane also never rips or tears, so we don't get the device failure associated with previous ones. And the idea is to use goose blood along with hen IgY from egg yolks to look at the efficacy of novel vaccine products. We have a bachelor flock of geese at Moredun Research Institute that we've been keeping for over 10 years. The geese act as blood donors for use in different in vitro diagnostic assays. And we found through preliminary work that mites are quite happy to feed on goose blood. So first I will optimise IgY extraction from egg yolk for use with mites, as obviously we don't want to use anything that might be toxic to them. We'll then use egg yolk extracted from extracted IgY in fresh and defrosted goose blood. We have a number of egg samples collected through previous vaccine trials. We already have the mortality and fecundity rates for those when they were used with the in vivo device. If successful, this will be a non-invasive way of testing novel vaccines in chickens. The larger replicate number, which is facilitated by the larger bleed volume in geese, also allows for additional research. So we can look at fed mites and use RNA extraction techniques to look at examine vaccine effects on mites and effects on mites through repeated feeding. I would just like to thank the NC3Rs for all their support and funding, Al Nisbet and Catherine Bartley, who have been working on poultry red mite for over a decade now at the Moredun, and Javier and Giles at BIOS for their statistical help, and the Moredun Bioservices team for their expert care of all the animals. Dan and Jose also, and our partners at Glenrath Farm and Bonington Farm, who are always supportive of all of our work. Thank you.
Thank you very much. It's really an honor to receive this prize and to have the opportunity to tell you about our research. And the title of the publication that was awarded is Pluripotent State Transitions Coordinate Morphogenesis in Mouse and Human Embryos. This sounds like quite a complex title, but there are basically two key messages that I would like to explain today. One is pluripotent stem cells. We have the term pluripotent in the title. Pluripotent stem cells are cells that are able to form all of the cell types in the organism. Bone, muscle, skin, every single cell type, those pluripotent stem cells can make. And we find those pluripotent stem cells in the embryo, at the time the embryo implants in the maternal uterus. You can see them actually here in this image of a human embryo, and they are labeled in magenta. Those cells will make the entire organism. And then there is a second term that I would like to explain, and this is morphogenesis. So morphogenesis refers to the formation of tissues and organs with a defined shape. And when I'm talking about morphogenesis here, I'm talking about the formation of this very small cavity that you see in the center of those pluripotent stem cells. This very small cavity will develop to form the amniotic cavity. This is a fundamental cavity during pregnancy that protects the developing fetus from mechanical damage and also from the immune response of the mother. So these are the two main elements in my research. And actually, the question that we were trying to address in this work was, what are the mechanisms that regulate amniotic cavity formation? And as I told you, this cavity is formed when the embryo implants in the maternal uterus. This happens in day 5 in mouse embryos, day 7 in human embryos. And these are just schematic representations of how the embryos look like at these stages. And I hope you can appreciate that when the embryo implants, it undergoes a complete remodeling of its shape. So this would be the mouse embryo. It forms this kind of cylindrical structure, and this would be the mouse embryo. The embryo looks completely different before and after implantation. And actually, mouse and human embryos look very different also as soon as they implant in the maternal uterus. But there is something in common in both species, and this is the formation of the cavity that I just mentioned, the amniotic cavity. You can see it here in the mouse embryo and in the human embryo. And what we discovered in this work was that actually those stem cells that will form the entire organism, which you can see here in red, those stem cells that initially are pluripotent, they need to lose this feature. They need to lose their pluripotency, and they need to start differentiation. In other words, they are not able anymore to form all the cell types in the organism. And this loss of pluripotency is necessary to make the amniotic cavity. Why is that? Because when the cells lose pluripotency, they undergo changes in gene expression, and this leads to the expression of proteins that are needed to build a cavity. And therefore, we end up having the amniotic cavity. Why is this important? Well, because we know if the amniotic cavity is not formed, development cannot progress. So alterations in amniotic cavity formation would lead to pregnancy loss. And actually, we know that 30% of human embryos fail to develop during this specific stage, at the time of implantation and early post-implantation. So if they undergo any problem in the formation of the cavity, this would lead to failed development and early pregnancy loss. So how did we discover this? How did we discover that cells needed to lose their pluripotent features in order to build the amniotic cavity? Well, actually, implantation is a really difficult stage of development to study, because as soon as the embryo implants in the maternal uterus, we cannot access it anymore, we cannot see it anymore, and therefore we have to dissect it out of the uterine tissue. This is an example of such an embryo. This is a mouse embryo that we have recovered from the uterus of a pregnant female. We just dissected the embryo outside of the uterus. But as I just said, this is extremely difficult. The embryo is very small at this stage. And moreover, this involves the use of a lot of mice. So we wanted to use an alternative method that would allow us to look at the mechanisms of cavity formation without needing to use as many mice. And we achieved that reduction in the number of mice used for research by culturing in a special 3D matrix embryonic stem cells. And by providing the right type of environment, what you can see here is that those embryonic stem cells are able to form a central cavity. That cavity is the in vitro equivalent of the amniotic cavity. And therefore, we can use this simplified 3D culture of stem cells to look at how that cavity is made. So this is how we achieve reduction. But actually, we also achieved partial replacement because we were looking at the mechanisms of amniotic cavity formation using human embryos. We developed a system that allows us to culture human embryos in vitro beyond the time of implantation, which I said is day 7. So beyond day 7 and up to day 13, we can maintain these human embryos in vitro, and they survive, they grow, and you can see here, they start to form that amniotic cavity. So we can look into that process in the context of human development. And finally, we also achieved refinement. So in a typical experiment, what we would do is recover embryos from a pregnant mouse, culture them in vitro, potentially do some type of modifications, and transfer those embryos back to a recipient female. And this procedure, in many cases, involves surgery, which causes moderate pain to the animals. And to avoid this need to transfer the embryos back to a recipient female, what we did was basically to take the embryos in vitro and culture them in the lab without the need to transfer them in a special type of culture media, which allows the embryos to develop beyond implantation and to form these typical structures of post-implantation mouse embryos, just in vitro. So this is how we achieved refinement. And by combining these different types of methodologies, 3D cultures of embryonic stem cells, human embryo cultures, and in vitro mouse embryo cultures, we were able to identify how the amniotic cavity is formed during development. But maybe you are wondering, well, you are using human embryos, you want to understand the reasons behind pregnancy loss and how the amniotic cavity is formed. Why don't you just use human embryos and forget about mouse embryos? And the reason is that, at least until now, what we could do with those human embryos is basically observe them. So we can culture them in vitro. You see here the embryo at day 7, day 8, day 9, day 10. We can see how they grow. We can see the type of changes in shape that they undergo, but we cannot manipulate them. And this would be really critical to understand what is the function of specific genes and proteins to interfere with those genes and proteins. And this is exactly what I'm planning to do with this prize. The objective would be to develop a platform to study gene function in human embryos. I recently opened my own group at the MRC LMB in Cambridge, and this will be one of my key objectives. And if we succeed at developing that platform with the help of the 3 Rs, we could look at the mechanisms of human development at the time of implantation, just using human embryos without the need for using mouse embryos. And we could address 2 key questions. How does the embryo undergo this global morphological change in shape as it develops from pre to post-implantation, and why so many pregnancies fail at this stage? And with this, I would like to thank everyone involved in the work. This work really involved different types of expertise, and this is why it was possible. So first of all, I would like to thank Magda, my supervisor during my postdoctoral fellowship, and she was really a driving force in this work, everyone in the group that helped me. I would like to thank our clinical collaborators that allow us to work with human embryos, and of course all the patients that donated their surplus human embryos from treatment. I would like to thank Krista for her help with gene expression analysis, and researchers from the Sanger Institute in Cambridge who helped us also analyze the gene expression profiles of the embryos, all the sources of funding, and I would like to thank you all for your attention.
Well, thank you very much to Francesca and Marta for excellent presentations. There are many wonderful things about Zoom in terms of cutting the amount of travel and time we spent getting to meetings, but the only downside is there's no real way of giving you a group round of applause. But if you can assume that we're all virtually clapping for the excellent work that you've done, and very much deserve this award, and I hope it continues to be a springboard for great things in the future of your career, because clearly both papers are really just the start of a longer and greater opportunities in the 3 Rs for these technologies. So I'm going to ask Katie Bates if we have any questions, if you can pose them, and then hopefully Francesca and Marta are on hand to answer any questions that the audience have raised.
Hello, I just wanted to echo Kevin's congratulations. It's really, really great to hear these 2 pieces of work have won the prize. So we've had a few questions come through and people giving their applause. So I just wanted to let you know that they're all clapping at home. So I guess the first question goes to Marta. So this is kind of more of a general one. So what got you interested in this field of research in the first place?
Yeah, so well, during my PhD, I was studying stem cells, but in the context of skin. So not only we have embryonic stem cells, but also adult stem cells. But then I thought that if we really want to understand how stem cells decide their fate, decide whether they want to be muscle or they want to be bone or they want to be brain, we really have to look in the embryo. And this is why I decided to join the laboratory of Magdalena Zernica Getz, to really understand how stem cells decide their fate. And if we want to understand that in a human context and to have relevance for disease and pregnancy loss, I think the best is to look at human embryos directly.
And the next question for Francesca. So do you see your device being able to replace field trials entirely? What would be needed to accomplish this?
I don't know whether it would completely replace them. It would really depend, I think, on industry, whether or not they would be happy with that, or whether sort of stakeholders further down the line would be happy with that. But certainly, I think it's a really good start of having some idea before you move forward into a field trial situation. I don't know if the stats would support sort of a smaller field trial situation. If you had good results for the in vitro, that would be one for statisticians, but yeah.
Great, thank you. And coming back to Marta, so we've had a question. So what genes do you want to investigate first in your future studies of gene function in human embryos, and what made you select those genes?
So we know that in the mouse there is a transcription factor that is called OCT4, and this gene is really important to make the amniotic cavity. However, we don't know if that's the case also in the human embryo. So this is one of the first genes that I would explore, really to test whether this transcription factor is needed to make the amniotic cavity.
And I guess follow-on question, what do you expect the time limits are to culturing human embryos in this way?
Sorry, can you repeat that again?
Sorry. So what do you expect the time limits are to culturing human embryos in this way?
Oh yeah, so I mean there is a legal limit which is the day 14, so we can culture human embryos only up to day 14 and not later. But just to look at the events that I want to study, which is the formation of the amniotic cavity and this initial differentiation of the pluripotent cells, we can do that within the limits. So that would happen between day 7 and day 13.
And coming back to Francesca, so this is more about the translatability of the device. So are there kind of other situations, so similar mite infestations or parasitic infestations in other species that the device could be applicable to?
Yes, I think so. Sort of ticks for a good example. It might take some sort of fiddling about to get the right kind of size mesh in terms of aperture and thickness, but on the whole I think ticks tend to have slightly bigger mouth parts, so it might be a bit less fiddly, but yes, it does.
And in terms of different species, could it be applied to different animals beyond hens?
I don't see why not. Exactly. I mean, it could certainly be used on larger animals. And I think one of the issues with the hens is that obviously where to put it on a hen, and the thigh is probably the most convenient place, but larger animals would have more convenient places, I think. So yes.
Great. Going back to Marta, so we've had a question about uptake. So are there other labs that would benefit from using your in vitro models?
Yeah, definitely. So, I mean, we know already that so many in vitro fertilization clinics have adapted our system to culture human embryos, and they are exploring things that they couldn't explore before using that system. So that regarding the human embryo culture, and then the 3D cultures of the stem cells are very useful, not only to look at stem cell fate decisions, but also to look at morphogenesis and how cells change their shape and interact with the extracellular matrix. So this could be potentially interesting for a scientist looking into cell biology, stem cell biology, and tissue biology, so different aspects of biology.
Great. And so I guess follow-on question, Marta, is are there any potential barriers for someone that would be looking to switch from using animals to your in vitro approaches? Is there something to bear in mind if they'd like to make that transition?
Yeah, so, well, if we talk first about the 3D culture, so the ones that I presented only recapitulate the embryonic tissue. So we can take those embryonic cells, put them in vitro, and they will mimic that morphogenesis of the embryonic tissue, but we don't have the extraembryonic cells. So if someone is interested in looking at how embryonic and extraembryonic stem cells talk to each other, this wouldn't be possible with the 3D culture system that I was using, the 3D stem cell culture system. And the lab where I did my postdoctoral training now has developed those kind of systems in which the extraembryonic cells are added. So I think what is really important here is to have a clear question, scientific question in mind that you want to address, and then choose the best system. The best system may be just the 3D culture of the embryonic stem cells, but that may not be sufficient. You may need to look into the in vitro culture of the embryo. So it will all depend on what is the specific question.
Great, thank you. Back to Francesca. So it's a question about how do you immunize against a mite?
Well, there have been various candidates. So Moredun have done a lot of work over the years and sort of in rational vaccine design. So really looking at tick orthologs or looking at whole mite antigen, and then seeing what that gets you in terms of sort of reduction in mite populations, etc. It's not easy to produce an ectoparasite vaccine. But obviously, sort of given the industry and given that the hens also already have a vaccine sort of protocol very early in life, if we could get something that could fall in with that and provide long-lasting immunity, that would be what we're after. But yes, it's not that easy.
Great. Thank you. So we're back to Marta again. So we've had a question about whether you're working with any products that are animal derived, such as FBS, within your models.
Yes, the culture media has FBS. Some people in Magdalena Cernica Getz lab tried to get rid of it, and it was only possible towards the later stages of the culture. The initial stages required FBS, and this is because FBS is needed for the embryo to attach to the dish. This is regarding embryo culture. In our 3D stem cell culture, we don't use FBS. So the 3D stem cell culture is completely chemically defined.
Great. The question for both of you. So a member of the audience has asked, what do you think was the key elements that helped you to get this award? So I don't know who wants to go first.
I'll go first if you don't mind, Marta. I think that ours is a very obviously applied piece of science. We're lucky that we work with BIOS in Scotland, who are sort of statistical gurus and help us with study design, etc. But it was just a very simple but effective piece of sort of innovation, really, that was enabled to have a big impact for, you know, reduction in hens and refinement. And hopefully the study that we do after that with the IGY will also build on that. So maybe that was it.
For me, I think our paper was really an example of how we can use novel methodologies to look into a specific developmental window that is extremely difficult to study. So it's thanks to the development of all these new methodologies that we can actually start to address these type of questions that before was completely impossible. So I think it's really a powerful approach to combine all these 3D cultures, embryo cultures, to look at the window of implantation, which is on top of that associated with very high rates of pregnancy loss.
Okay, thank you. We've got a couple more questions that have come through. So if we go to Francesca again, so where did the idea of the on-hen device originate from in the first place?
That was a colleague, Kath Bartley, who has been working on red mite for a long time. So she was in the team before I joined, and I know that they tried sort of various different on-hen prototypes, both mostly rigid ones. But you can imagine with a rigid device on sort of a muscle, any muscle really, as the muscle moves, the rigid device moves with it, which means the mites can't attach and feed very well. So they came up with this flexible device that I spoke of that allows for better, if it's attached it's quite snugly, it allows for better mite feeding. And actually from that first prototype, it's changed a little bit in how we construct it, and also for ease of handling, but it hasn't changed that much apart from the mesh size since then.
Great, thank you. And I guess another follow-on question, Francesca, is, so could you tell us a bit more about the studies that are performed on the mites once they're recovered from the on-hen feeding devices?
Yes, so once the pouch is removed, we very carefully take it apart. Now, because the mites are so fast, we have to handle them on ice at all times to slow them down a little bit. So we take the fed mites and we put them into individual wells in sort of an ELISA plate, and we cover that with a porous membrane, and then we monitor them for, you know, every 2 days or however how often we like, to see when they die, when they produce eggs, when those eggs hatch, etc. And we keep them in an incubator which runs at sort of optimal conditions, which is 25 degrees centigrade and 85% relative humidity.
Thank you. And a question to both of you. So how are you both hoping to promote your 3Rs approaches and increase uptake in your fields?
Well, I think it's really critical to go to meetings, to talk to people. It may not be that possible these days with coronavirus, but at least using virtual meetings and just talking to people, and even using platforms such as Twitter to promote our research, I think is the best way.
There's a large research network of poultry red mite researchers across the world, but particularly in Europe. which we've been very involved with from the start. We have been really lucky, and we're really pleased to have this NC3R's Knowledge and Exchange grant, which we're going to hold a workshop hopefully. We've got lots of people interested in coming over and actually getting some sort of hands-on experience of how to make the pouches and handle mites. So we're looking forward to that, and hopefully they'll take that back to their different groups and take it up and employ it that way.
Thank you. And again, another question for both of you. So do you take inspiration from any other fields, or is there a field that you would like to collaborate with?
Well, I mean, I think probably the future for the kind of technologies that I describe is to really join efforts with bioengineers to develop more complex platforms that would allow the embryos to develop for longer and to mimic certain aspects of their development that we cannot mimic with our platforms. So yes, really talking to bioengineers, I think it would be a great way forward.
The tick field is very interesting. I think mostly I'm interested in the biology of the mites. We don't know much about these mites, weirdly, considering that they're such a huge problem, but also looking at sort of refinements in other animal disease models really interests me as well, to see if there sort of any scope for refinements in those as well. So kind of quite broad really, but no specific discipline other than veterinary. I'm very much involved in veterinary.
Thank you. So I don't have any more questions that have come through.
Okay, well, Katie, thank you very much for handling the questions so well, and thank you for all the hard work you did to make the prize possible and run so smoothly. I think we'll bring the prize ceremony to a close now. Let me just again congratulate the two winners. Thank everyone. We got well over 150 people attending. Thank you very much for attending and continuing to support the NC3Rs. We have recorded this, so we will be putting this up on the webpage. So if you want to view it again or tell other people about it, it'll be up there very shortly. So once again, thanks everybody. Congratulations to the winner, and we'll see you for future ceremonies.